Let's welcome Iakovos Lazaridis. Iakovos holds a degree in molecular biology, followed by master's degrees in immunology and, of course, brain and mind sciences. He earned his PhD at the University of Crete in 2011, then spent a decade at the Karolinska Institute before joining the McGovern Institute for Brain Research at MIT, where he currently works. Iakovos, the floor is yours.
Hey, thank you very much for the invitation and the opportunity to share my work. I will start by giving you a bit of the outline of my talk. I will start with a brief introduction about basal ganglia and dopamine function, mainly to frame the questions that we will deal with or inspired the two main parts of the talk that are two very different mechanisms that can be involved in the control, in the regulation of dopamine feedback to the striatum, with one being a pathway-specific pathway from striosomes directly to dopamine that form a feedback loop, and another that is a volume-based local feedback loop that involves interaction between non-neuronal cell type astrocytes and dopamine. So to start with, basal ganglia sit in the center of action learning, and this involves several processes that are happening at the same time. At the higher level, you have decision making. You have to find your goals and pursue your goals. And at the second level, you have motor control. The system has to prepare and execute and refine the actions that will bring you closer to the goals. And importantly, all of this is happening in a dynamic environment. So the system is not only learning skills, but it has to remain flexible. It has to be able to stop or suppress previously learned actions when they're not no more appropriate, and to learn new ones when the contingencies change. Now, dopamine is usually introduced here as a teaching signal involved in reinforcement, outcome evaluation, reward prediction errors, and clearly this is one of the main roles of dopamine. However, already you can see that a single unified dopamine signal cannot realistically deal with all these different processes that are happening at different time scales and temporal windows. And maybe a way to visualize this is, and to illustrate this is going from a scalar teaching signal that just scales in amplitude into potentially a channel-weighted teaching signal that contains components of the different circuits. And we do have also information from the architecture of the system. We know that the basal ganglia are structured in closed cortico-basal ganglia-thalamo-cortical loops with information from different areas in the cortex having a specific topography on how they innervate the input area of the basal ganglia, the striatum. And we know that this topography continues throughout the basal ganglia and into the thalamus and back into the cortex, forming these parallel information channels, these closed loops of parallel information channels. So this architecture suggests specific requirements. This means that you have distinct circuits deal with different information, and you will need a specific feedback information also to entrain these loops. Also, we know the dopamine system itself is not made from homogeneous dopamine population. We know that dopamine neurons are segregated both in location, in molecular identity, and projection patterns. We know that they have specific projections to distinct parts of the striatum and associated with distinct different functions. So the two mechanisms that I will show you are positioned exactly in dealing with this, providing the information of that a local circuit, a local pathway will need into the dopamine neurons in order to shape the feedback to be more meaningful for this particular function. So I will start with the projection-specific pathway that involves a striosomal feedback loop. And in order to do that, I will just give you some more information about the basal ganglia structure. So as we said, the information from the cortex enters the basal ganglia through the striatum. And then the information continues downstream through two main pathways. The direct pathway, or as known as a GO pathway, that is carried out by D1 expressing medium spiny neurons. And the direct is referring to that is directly targeting the output nucleus of the basal ganglia, the globus pallidus internal, and the substantia nigra reticulata. And the opposite is happening from the indirect or no-go pathway, in where it targets these output nucleuses indirectly through the external part of globus pallidus. Now, doing that, and the reason they're called go and no-go pathways is that the D1 pathway is inhibiting the output nucleuses that normally are tonically active and inhibitory, and they work like a door, keeping closed the downstream motor commands. So as long as the output nucleus of the basal ganglia, the SNR and the GPI are active, then you block commands from the cortex to reach the execution centers. And D1 pathway, the GO pathway, by inhibiting these centers, it allows the actions to these commands to reach to these centers. The opposite is happening from the indirect pathway, where it disinhibits the center by inhibiting the external part of globus pallidus, and that means that it strengthens this suppression that you have in the execution centers. Now, except from these two main pathways, also I have to say that dopamine is playing a very important role here by biasing the excitability of these two neurons and biasing it in a way that is favoring D1 activity and suppressing D2 activity. And by doing that, it's playing a very important role in learning through regulating the cortico-striatal connection. Now, except from these pathways, the striatum is also divided into different compartments, the striosomal and the matrix compartment. And these two compartments, they don't have clear anatomical boundaries, and also you cannot separate them by just the distribution of D1 or D2 neurons, but they represent distinct functional domains. Now, the reason that we started looking at these and trying to find if striosomal pathway can provide some information, dopamine or some local modular information, is because we had information both from Anne Grabio's lab, previous work, and from others, that at least part of the striosomal D1 direct pathway has contacts directly onto dopamine neurons, specifically into the distal dendrites extending within the SNR, forming this characteristic dendron bouquet structure. But what we didn't know is the extent of this connection, if this is just some collaterals, or if this is the main pathway of the striosomes, and also there was no information about the potential indirect pathway, striosomal pathway, that can affect dopamine. So we started by trying, maybe, can I remove this? Yeah. We started by wanting to map all the inputs into dopamine neurons in the SNC. And in order to do that, we use a dual viral system with a rabies virus. And in this system, you introduce the rabies virus specifically targeting genetically identified neurons by using the helper virus. So the helper virus is a CRE-dependent virus that will express the required proteins that the rabies needs in order to both infect the starter cells, but also to jump one synapse back and labeled all the input neurons in the entire brain that have contact to these starter cells. So with the helper virus, we express the G protein and a TVA, that's the receptor that has been modified on the rabies virus with an NVA to recognize. So by using a DAT-CRE mice, we can express the TVA receptor and the G protein specifically only into dopamine neurons within the substantia nigra compacta. And then by injecting the rabies virus in the same place, then we ensure that we infect only dopamine neurons within that area. And because these starter neurons also contain the G protein that the rabies requires to jump to the postsynaptic cell, to the presynaptic cell, then we know that we will label all the cells that have direct synaptic contact. And since these cells, these input cells, do not contain the G protein, the rabies will not continue. So we have only the monosynaptic connectivity. So by doing that, we can also see how specific is that. We can count and identify how many are the starter cells, the dopamine starter cells. So then we can really understand and make calculations depending on the position and the number of the starting population. So by doing that now, you can see here everything, all the green cells that you see in the entire brain are cells that have a direct synaptic contact onto dopamine neurons within the SNC. So the first thing that we saw and that we're kind of expecting from previous information is that we saw that the majority of cells that have direct contact with dopamine neurons in the striatum live within the striosomes. And this is around 70 to 80% of the neurons that you find in the striatum live within the striosomes. What we didn't expect is that we also found this very specific pattern within the external part of globus pallidus, with, as you can see here, the majority of the cells living in the central zone, that we named it like that after seeing it, and avoiding the border regions of the GP. And again, you can see that this is pretty high specificity also for that. So now that shows a very specific striosomal direct pathway, but it gives the potential that there is also a potential indirect pathway that starts potentially from D2 striosomal neurons and target a specific population and area within the GP. Now, in order to see how specific this pattern, we did the exact same experiments, but now instead of starting from the dopamine neurons in the SNC, we went into a PV-CRE line that we know that PV represents the majority of neurons within the output nucleus of the basal ganglia, the substantia nigra reticulata, and repeat the same experiment. So finding all the input neurons in the brain that target the output nucleus, the motor part of the basal ganglia. So by doing that, surprisingly, we find that it's completely complementary. So first of all, looking in the striatum, we didn't expect that it's going to be that exclusive, because that means now that is not that striosomal neurons contact also dopamine, but this pattern shows that this pathway is more exclusive. It looks like the majority of striosomal neurons, they do not actually target the output nucleus of the basal ganglia. That makes it a specialized pathway to control dopamine. And the same applies for the central part of the GP. And you can see how complementary again is this pattern. The neurons that within the GP that project into the SNR live in the border regions, avoiding the central zone that is occupied by neurons that control the target dopamine. So now this looks like a very exclusive and specific pathways from striosomes and from the GP. And what is missing is if this central region is actually connected to specifically to striosomal D2 neurons. So in order to do that, we repeat this tracing experiments, but now we did this by crossing a striosomal line that will express TD tomato in striosomal neurons and that create to target the dopamine neurons. So now by doing the same tracing, we can label the neurons that project directly onto dopamine and have at the same time the terminals that come from the striosomes within the GP. And by doing that, we saw that these terminals are also associated in the central zone, and you can find very close to neurons in the GP that they're labeled retrogradely because they target dopamine neurons, that you find a lot of boutons associated with them. And the majority of them are also in the dendrites, as is the usual connectivity from the striatum. And we went also when we stained for axial terminals with synaptophysin and other markers, and we did show that the majority of the striosomal terminals actually are in contact with these dopamine projecting GP neurons. So just by looking this anatomy, it looks like there is a parallel striosomal pathway that is exclusively there to control, to send information into dopamine neurons. That makes it a possible candidate, the striosomes, to be there specifically to provide the information that its module within the striatum needs into dopamine neurons to shape appropriately the feedback of dopamine. So, okay, we do have this anatomy, but the question is what is the functional role of this and how it works. So in order to do that, we want to do optogenetics by using two of our striosomal lines, D1 striosomal line, that is the nociceptin CRE line, and the neurotensin line that is labeling specifically D2 striosomal neurons. And by expressing crimson opsin, they're an excitatory opsin, and inserting a fiber within the dorsal medial and dorsolateral striatum, we ask the question, and also at the same time expressing a sensor for dopamine, was the question how the activation of these two pathways, the direct and indirect stereosomal pathways, can affect dopamine. And what we saw is that, as the anatomy suggests, because these are all inhibitory connections, the D1 direct connection you would expect that will inhibit dopamine, and the opposite you expect also that the D2 indirect pathway will do. And this is exactly what we find, that is suggested by anatomy, but is a little bit surprising knowing, because it's against the classical go-no-go pathways from the matrix. So the question is, what is the behavior effect on that? So while we were doing these stimulations, we were recording the behavior of the mice, so we're able to see what these effects are. And the first thing that we would see, just by labeling the distance traveled before the stimulation, during the stimulation, and after the stimulation, is that, again, as you expect, when you stimulate the D1 striosomal pathway that inhibits dopamine, it also reduces mobility of the mice, reduces this trajectory in freely moving mice, and the opposite is happening when you stimulate the D2. And we went a little bit further. We did the behavioral clustering, and we also saw that the effects are opposing each other of D1 and D2. So the first thing that you see here is that this contradicts the classical direct and indirect pathway. It looks like it has the exact opposite effects, with D1 now being the no-go pathway and D2 pathway starting from striosomes being the go pathway. So this is the final, just the summary of this part of the work. We did so that the striosomal pathway forms a parallel pathway that is specialized in the control of dopamine feedback into the striatum. And this can provide then the means for giving this channel-specific, channel and state-specific training entrainment of the dopamine signals back into the striatum. Because maybe I didn't say before, but the structure is we think that there is a modular architecture in there. You have a part of striosomes and a part of matrix associated with the same kind of similar information. So by having the striosomes providing this information, then you can shape within the same vector in the required function to specifically modulate the matrix pathway. Okay, and moving to the second part that involves this non-neural type, the astrocytes and interaction and feedback loop with dopamine. Again, I will need to introduce a little bit the astrocytes. So astrocytes, till recently, they were considered housekeeping cells, getting involved in controlling the blood-brain barrier and supporting neurons in several ways, including also reuptake, clearance of neurotransmitters, and we also knew that they can release also these neurotransmitters that they reuptake, including other factors, and can also modulate the synaptic activity. When things start becoming more interesting is when we realize that these processes are dynamic processes controlled by calcium signaling within the astrocytes. So astrocytes, by sensing, by reuptaking the neurotransmitters, these at the microdomains, where they engulf the synaptic space, this affects the calcium image locally within the microdomains. But there is something like a threshold. People do not like to call it the threshold, but when the activity around the astrocyte is listening is somehow getting a lot, then you reach to an active process, or that is a release from endoplastic reticulum of calcium that reaches to a very strong response that you find a signal that reaches to the cell body and is not staying only there because the astrocytes are connected with gamma junctions. This can expand into neighboring astrocytes, covering a very large area of the network. And it became even more interesting when we realized that this calcium signal is also affected by the neuromodulators of the brain, and mainly from work in the cortex and norepinephrine. So now you have a non-neuronal cell type that it modulates dynamically reuptake and release of neurotransmitters, integrating both local activity and the neuromodulators of the brain. That makes it very interesting to see if they're actively playing a role in the computation of the network. So the first thing that we want to see is to see if there is any relation between dopamine and astrocytes. And in order to do that, we recorded in freely moving mice with calcium imaging in both dorsal-mediated and dorsolateral striatum simultaneously, dopamine by using a dopamine sensor and astrocyte calcium activity. And we did that within a decision-making task, two-choice probabilistic switching task, because we knew that we can get a lot of information there, both from kinematics, reward or outcome responses, and action values, and other things that we know that dopamine usually is encoding for. So before we go within the task structure, the first thing that we notice by doing that is, and here I just show you a small snippet from a single recording of simultaneously recording astrocytes and dopamine signals in the same animal, is that whenever you will see a large peak of dopamine concentration increase, this is usually followed by an astrocytic response. And in order to see if this holds as a more general principle, we took the recordings from all the sessions and all the mice freely moving and performing the task. And we just make a peak triggered averaging. And what you can see is that if you plot the average signal of dopamine in dorsal-mediated and dorsolateral striatum aligned to the peak of astrocytes, you find that this peak that is happening exactly before the astrocytic peak. And if you do the exact opposite, you align the calcium signal of astrocytes to the peak of dopamine, you find that the peak of astrocytes usually happens very close after the dopamine peak. So now this shows the exact same relation that you just see by looking the raw signal, but again is restricted only to these large peaks that we picked. So we went and did a more unbiased analysis by doing a cross correlation using again the whole signal now. And here is where we saw again the same relation. You have a positive correlation between the two signals with a lag at around 2 seconds. You have a positive correlation from dopamine peak. But also here you can more easily reveal that there's also a correlation in the opposite direction that is around much slower, is around 8 seconds after the astrocyte peak, you find a suppression, the peak of a suppression in dopamine. So this tells you that it can be a closed feedback loop with dopamine directly or indirectly activating, inducing calcium signals in astrocytes, and then this response in astrocytes somehow it affects the dopamine levels, either from clearance or affecting release from the terminus. So we will come back to that relation, but let's go a little bit now within the task. So even though that we find this close relation between the dopamine concentration and the calcium signals in the astrocytes, when we looked within the task structure, we saw that the astrocytes within the task, and this normalized time from initiation of the task in the central arm, running the central corridor, the turn, and the choice board, we find a moderate modulation of the astrocytes compared to dopamine. But still, this was inverted. So you have usually the dopamine is low within the ports, and it increases while the mice is running in the corridor, and is the opposite direction of the astrocyte, but with reduced amplitude modulation. And also, when we looked now about outcome responses, again, we saw that there is a very moderate, there is no information in the astrocytes about the outcome. So you cannot separate by looking astrocyte calcium signal if the trial was rewarded or non-rewarded, or extracting any kind of value of the action. Things that you can do by using the dopamine signal. And this is a bit surprising because we saw this very strong relation between the two signals. So we didn't see any outcome evaluation, any outcome information on the astrocytes, but what we noticed, again, by looking at the raw signals, is that you find, this is an entire session, you find these time points where you have these very strong, very large in amplitude peaks in astrocyte calcium signals. And just by looking again the structure of the task, here plotted the initiation, so the initiation of each trial, you can see that usually this happens when the mice is not engaged in the task, is not performing the task. And in order to better quantify that, we made an initiation day function that is representing the engagement of the mice. And what we notice is that usually this peaks happens during disengaged periods, but very close to the next engagement, re-engagement. And when we see, so collectively that within all the mice and all the sessions, we saw that this is true also for dopamine. So you have these large peaks associated just before the next re-engagement. And again, to see this a little bit better, we identified all the engaged periods and we went backwards. So this is engagement based on our definition. And we went backwards and find the first initiation, the first trial initiated that start affecting this initiation density function. And what we found is that dopamine signal is associated very closely to this first trial that was initiated and follows the astrocytes signal also. So just by looking at that, it looks like it's possible maybe by looking the signal of astrocytes, the calcium signal of astrocytes, to maybe predict the engagement state or the upcoming engagement state of the mice. So to do that, we again clustered the behavioral, the kinematics of the mice, and we manually labeled different kinematic clusters on task-related behavioral snippets and non-task related. And for instance, an example here is that you can see here is grooming, and this cluster will go in a task unrelated, while engagement with the either initiation port or the choice ports or running in the corridor towards the port are kinematics associated with the task. So just by doing that division, then first we went and we looked based on our own definition of engagement, and we asked the question, what is the ratio of usage of these different clusters? And as you can see, if you align this into the peak of engagement, then you find that you have increased uses of task-related behaviors and a decrease of behaviors that are not unrelated with that involves grooming, rearing, and immobility. So we did exactly the same by aligning that to the astrocyte peak. And as you can see, the astrocytes gives a very similar picture. So that means that just by looking at the astrocyte activity, you can predict if the mice is going to be engaged in the task. And to do this in a little bit better way, we use the classifier and trying to ask what is the accuracy of dopamine or astrocytes, both for task events and engagement states. And what we find is that while dopamine is performing well in both of the task space, from initiation to also dissociating rewarded and rewarded, dissociating different outcomes, and also engagement states, it looks like astrocytes, as we saw before, they're not that good in predicting outcomes within the task space, but they look that they are pretty good in predicting engagement states, and specifically the engagement state and the transition from unengaged to engaged. So what I show you till now is that by looking the calcium activity of a non-neuronal cell type, the astrocytes, you can potentially predict a behavioral state. So there are several questions that come with that. And one of them is what is the nature of that relation between dopamine and astrocytes? Is this relation that we find with astrocytes and behavioral states a causal relation, or just reflects potentially dopamine or other network changes? And if it is a causal relation, what is mediating this potential change in the network state and potentially the behavior? So we will start from the question if it is causal, because it is an important one to also see how interesting it is to continue pursuing that. So to do this, we went and we suppressed calcium signals in the astrocyte by using this PMCA, that is a calcium pump, is an ATP pump. And what is the effect by expressing this in the membrane of the astrocytes is that it's constantly pumping out calcium, and in a dynamic way. So the higher the concentration of calcium, the more calcium it pumps out. So you end up with a flat calcium signal in the astrocyte. So you block the calcium transient in astrocytes. So by injecting this bilaterally in the dorsal striatum in mice, then we went specifically to an auditory discrimination task. And we did this because we know that in this task, we can dissociate in different axes engagement from discrimination learning. So the way that this task is working is that you have two different cues, tone cues. One is representing a rewarded outcome, where the magnitude of it is dependent on the response rate. And the second cue is associated with the cost cue, in where you get an aversive, the mice will get, if they respond, they will get an aversive stimuli that is an intense light, that again, the intensity is dependent on the response rate. So as the mice learn the task, they learn to accept the trials that are associated with the rewarded cues, and to reject the trials with not responding when the aversive cue comes. So by now looking at the behavior of the control mice and the mice that have suppressed calcium signals in the astrocytes, what we notice is that there is a difference that is specifically during early learning. And we know that this is a specific engagement a deficit, because in this plot, when you plot cost, the acceptance rate of cost trials against the acceptance rate of rewarded trials, then you can form these two axes. The higher in the upper right of this plot you are, that means that the more engaged you are, and the higher on the left up side you are, that means that you have a better, higher learning, discrimination learning. And you can form then these arcs in where you, as long as you live within this arc, you are within the same learning, within the same discrimination learning, but you have different engagement states. And the same applies within these arcs, but in the opposite way. While you're living in here, you have the same engagement state, but different learning. And just to maybe give you a better understanding of that, if you live, for instance, at this point, that means that your total acceptance rate is high, but also your discrimination learning is high, since you mostly accept the rewarded trials, and you reject the trials that are associated with the punishment. While if you live here, again, your response rate, your engagement, your total engagement is at the same level, but now your discrimination learning is pretty bad. You cannot discriminate what is the rewarded or unrewarded cues. So what we saw here is that during the early learning, the mice that have suppressed calcium signals in astrocytes, they saw a specific deficit that is in engagement. And this goes away in late stages of learning. However, now we're still running this is an ongoing experiment, and we want to see when the plateau would be reached, because these effects during early learning can potentially affect the total performance. But at least at this point, we find an effect that is specifically in engagement, and it's happening during early learning. And we know that this is not a generalized effect, because the mice cannot, they perform their leak rate or any other motor effects, because if you go and look within the accepted trials, then they have a very similar performance. And we also know that this is not a motivation deficit, because we also run an effort-based breaking point paradigm, in where the mice have to leak to get reward, and this number of leak requirement is increasing exponentially with every 6 blocks of trials. So you reach to a point after a certain level where the requirement of the leaks is that high that the mice stop performing the task. And this is called the breaking point. And this task is specifically designed to see the potential motivational effects. And we found no difference between the PMCA mice with suppressed calcium signals and control. So taking together this, it looks like there is a potential causal relation between astrocytes and behavioral state, since we had specific engagement deficits during early learning when we suppressed the calcium signals. That is also aligns with our initial observation, where we saw that these large signals and the predictive power of the astrocytic signals for specifically engagement states. So since now we saw that you can actually, astrocytes are in a position to affect potentially network state and behavioral state, we went forward to try to understand what is the mechanism. And again, the first thing that we wanted to see a direct relation of dopamine and astrocytes. We went in DAT cream mice and expressed an excitatory opsin in the substantia nigra compacta in order to stimulate specifically dopamine neurons. And then we wanted to record astrocyte calcium signals within the striatum. But before we do that, we recorded dopamine to verify that actually this substantia nigra compacta stimulation, it actually induces dopamine release in the striatum. And we did see that there is a very robust release with every stimulation train. And since we saw that, then we went to the exact same stimulation protocol, but this time recording calcium signals from astrocytes. And what we found is that this stimulation in substantia nigra induces a very strong response of astrocytes. And it's also interesting to see the timing of that. Dopamine release starts very close to the start of the stimulation train. This is a 4-second, 40 hertz stimulation train. And starts recovering immediately after the stimulation ends, while the astrocytes have a delayed start and they peaked after the stimulation train has ended. Now you can also see here that there are some difference between the two signals. You see that the reduction in dopamine from trial to trial at this in the trial interval is much smaller from what you can see in the astrocyte. So we want to measure this. So the first thing to tell you is that both of them look to be have a frequency to be frequency dependent. So the higher the frequency that you stimulate the dopamine neurons, the more dopamine you release. And it looks like astrocytes also follow that. The higher the concentration of dopamine, the higher the response of astrocytes. However, there is a non-linearity that we saw here. While the frequency response of dopamine, it looks very close to linear, then you have this almost exponential or much sharper slope in the response of astrocytes that looks like they ignore small concentrations of dopamine, and there is a level of dopamine where they have a very sharp increase in their response. We also found a similar nonlinearity with an adaptation in a much stronger adaptation in the astrocyte responses from trial to trial. Again, you can see that there is a small adaptation in dopamine, but there is a very large adaptation that happens after the first response of the astrocytes. And this drives us to do intra-pulse interval. This is actually an intertrain interval in where we wanted to see if there is a specific refractory period in where the astrocytes cannot respond with the same amplitude as the first response. So to do that, we made an initial stimulation train of the substantia nigra compacta, and we gave with a different interval a second train. And what we found is that there is a refractory period that has the peak of suppression around 15 seconds after the first train. And the period of this refractory period is pretty long. You have to go to almost 2 minutes after the initial strong dopamine release in order to get the recovered levels, the same amplitude response in the astrocytes. And this is a little bit mechanistic, but it's going to be interesting a little bit later. So now this shows a direct relation. By inducing dopamine, you get a clean response in the astrocytes that has these characteristics, but still doesn't answer the question if this is a direct relation or this is potentially an indirect through the effects of dopamine in the local neural network that then this activity is being picked from the astrocytes. So to answer this question, we went and we specifically knocked down dopamine receptors and norepinephrine receptor, because we know that there is also an affinity of dopamine for this receptor. It's 100-fold lower, but since we know this relation from the cortex, we also want to see if it plays a role. And what we found is that only when we specifically knocked down the expression of D2 receptor, specifically only from astrocytes, we're able to almost completely abolish this response. So this shows that potentially this relation is direct. Dopamine acts specifically on D2 receptors on astrocytes to induce these calcium responses. Now we will go for a little bit. We saw from the cross-correlation that there was also an effect on dopamine levels from the astrocyte responses. And again, to find the causal relation of that, we went and we stimulated, we induced calcium signals in astrocytes genetically, and we recorded at the same time dopamine. And what we saw is that you get this strong suppression, reduction of dopamine concentration during the stimulation of the astrocytes. This is a pretty long stimulation. It's a 10-second stimulation trying to mimic this long, extended calcium signal pattern that astrocytes have. And also it's interesting that you can see that there is a very slow recovery of the dopamine levels that again takes close to a minute to come back. So coming now to summarize this part, we did show that there is this relation of dopamine to astrocytes is direct. Dopamine acts on to specifically D2 receptors on astrocytes, inducing these calcium signals, and this calcium activity in astrocytes can reduce the dopamine levels. So again, this raises a question: how is this reduction being mediated? That adds on top of this question, again, what is the mediator that can affect the neuronal network state and behavioral state? So the first candidate that we wanted to test is adenosine, because it is known that astrocyte can release ATP that extracellularly then is modified, is metabolized to AMP and then into adenosine from enzymes that live on the membrane of the astrocytes. So we did again the same protocol of stimulation, but this time we recorded adenosine in the dorsal striatum. And what we find is that indeed this stimulation, this dopamine of the genetically induced dopamine increase in the striatum results in a strong increase in adenosine levels. And just to be able to compare the time course of these with dopamine and astrocyte response, you can see now that with blue you have dopamine, in red the astrocytes, and in green the adenosine. You can see, if you just see the timing of the peak, the first to peak is the dopamine, then follows astrocytes, and then a bit later follows the adenosine. What is more interesting is the decay time. So you can see that adenosine has a very low decay time that reaches almost a second, and you can appreciate that more if you see the whole trace. And also here is important to say that this adenosine release, it is astrocyte dependent, because if you block calcium signals, again by using the PMCA pump in the astrocytes, you can block also the adenosine release by the adenosine release from the optogenetically induced increase in dopamine concentration. Now, it's interesting, as I told you before, I show you a little bit of the some mechanistic things, because it becomes interesting. This timing, this slow decay, can potentially explain both the slow recovery of dopamine levels, but also the refractory period in the astrocytes. It looks like as long as the adenosine levels are high, then you have a lower dopamine, and also you have reduced response to dopamine of the astrocytes. So just by again summarizing this part, we did show here that you have a potential candidate as the mediator of both potentially the dopamine suppression and the network activity. And this is because, and maybe I will summarize it better here, is because we do know that there is inhibitory adenosine receptors onto the terminals of dopamine neurons that can potentially mediate this suppression. And also, even more interesting is that you have differential expression of A1 and A2A on D1 and D2 neurons, with A1 that is inhibitory being a little bit more broadly expressed in both of D1 and D2, but then you have an exclusive expression of the excitatory A2A only in D2 receptors. And that means that adenosine can also potentially affect the balance between the D1, D2, and the go, no go pathway. And just to show you a hypothetical time course of this, is you have the dopamine release, this induces astrocyte response, adenosine release, and then you go from a go favoring balance that the dopamine induces to a prolonged window that can act as a reset window that suppresses actions, reducing the noise for the state to change, doing a new behavior. And we are right now in the process of directly linking the production of adenosine and adenosine itself and its action on the receptors in dopamine terminals, and specifically also in the D2, D1, to see if it's actually able to balance, to change the balance between the direct and indirect pathways. So to summarize both of these, the entire talk, I did show you two complementary mechanisms that they're both positioned in the place to be able to provide local information into dopamine, one by having a very specific striosomal pathway that can give information modular and information channel specific information onto dopamine neurons, and the local feedback between astrocytes and dopamine that can locally regulate both dopamine tone, but also potentially affect the balance between the go-no-go pathway. And both can result, can go from these scalar signal now to shape dopamine into a channel-weighted teaching signal that contains components of the vector of the specific function of this circuit. So I want to thank you all and all my colleagues that have participated in this work. Thank you very much.